All right. Thanks everyone for joining us today. It is my pleasure to introduce Alan. Alan is an associate professor in the Department of Applied and Computational Math and Statistics at University of Notre Dame. His research focuses on using computational and analytical techniques to study PDEs arising in modeling of physical and biological systems. So today he will share with us some of the related work about mathematics of diffusive transport with applications to chemoreception and nuclear scaling.
Well, thank you, Ying, and thank you for the opportunity to speak a little bit about my research. And someone was asking about this picture here, and what it is the stigma of a flower with pollen on it. And this image is curious to me because it's characteristic of a lot of phenomena in biology where there's something very essential happening, which is the reproduction of the flower, yet the pollen is being dispersed by complete random forces, yet somehow everything kind of works out. So generally the theme of this research is kind of understanding how these essential processes, while they're completely random, they all seem to work predictably, and often the stochastic effects are constructive rather than just a nuisance to be overcome. So let me first of all acknowledge my collaborators. I've worked on these problems the last 4 or 5 years or so, and some of my longtime collaborators are Andrew Burnoff at Harvey Mudd, Theo Kollenkoff at Dalhousie, Michael Ward, who's my PhD advisor at the University of British Columbia, Ryan Spoonmore is an undergrad who's worked on these problems, Daniel Schmidt is an undergrad who worked with Andy at Harvey Mudd. I've been working with Chris Miles and Sean Lawley. We'll be talking about some of the results there. And some other collaborators at Notre Dame, Jacob and Samuel were undergrads who worked with me. Benzu is a postdoc, and Adrian Navarro is a current PhD student. And at the end, hopefully I'll talk about some ongoing work with Angelica Manhart at UCL and our experimental collaborator, Jay Gatlin at University of Wyoming. So I'm kind of interested in diffusive transport. So how do things get transported on cellular scales using purely Brownian motion? And as I'll talk about a little bit beginning, the first part will just be some recap on some basic things about Brownian motion and some of the key results that will be important later. But really what's essential is the geometry, whether your domain that you're considering the diffusive process, is it bounded or is it unbounded, and the spatial dimension. So on the left-hand side, you see a situation where it's really an annulus. There's an outer disk and then an inner disk. And there's 4 Brownian random walkers going on here. And the process ends when they reach this inner annulus here. And you can see this one reaches very quickly, and these will take longer to get there. And there's a similar picture on the right, except that now it's in an unbounded domain. So I don't think you'll see this, but if any of these particles reach the outer boundary, they'd be reflected and they'd have to come back in. That makes a big difference in the characterization of the random walk, whether it's in a bounded or unbounded domain. On the right-hand picture, it's a 3-dimensional scenario, exterior to the sphere. And again, you see 4 particles and they're trying to reach the sphere, but moreover, they're trying to reach certain locations on the sphere where reactions may occur. So some of the sort of questions that are kind of always present is, will the diffusing particle reach the target? Will it get there at all? And if so, how long do you have to wait for that to happen? If the target sites which they have to reach are they mobile? Are they fragmented? What's their distribution? How does that configuration play a role in the first question, whether they find the target and the time? So let me talk about 2 applications that are motivated by specifically. And one is the nucleus. Okay, so on the right here, we see a picture of the nucleus. I think it's a human cell. And transport across the nucleus is very highly regulated. There's genetic material inside that needs to be processed, and there's a lot of things going back and forth. And so the nucleus is generally an envelope, and the gatekeepers of the material that can go in and out are the so-called nuclear pore complexes. And what they are roughly 2,000 surface pores they occupy a very small portion of the surface. And you can see, I zoom in here, their combined area is about 2%, but the number in the thousands. So how do we even characterize, one of the things I'm interested in, how do we characterize transport over such a membrane? So it's a very small fraction area, but many, many openings. Now the process of getting in and out of the nuclear pore is very complex. So you can imagine a particle could diffuse towards it, reach the nuclear pore complex, and then get in. They're actually not simply a hole. They have more sophisticated mechanisms to ensure that particles or molecules can only get inside when they're in the correct conformal state. So it might mean that they have to have undergone some reaction before they reach the NPC. And if they're in the correct state, then they can enter into the nucleus. So there is some more sophisticated stuff going on here that many people have thought about and modeling of. Another application that been motivated by recently is yeast mating. So before mating starts, the receptors on the surface of the cell are uniform. And of course, there's chemical signaling between each of these cells. They release pheromones, those pheromones diffuse, and eventually they'll alight on the receptors. And once they do, they start clustering. So the initially uniform profile starts clustering in the direction of the target cell. And so how do we understand this mechanism of receptor clustering? In the case of the nucleus, they're actually quite uniform, the configuration of pores. And I will say sometimes I use the word pore, sometimes I use the word receptor, and I'm using them interchangeably because generally it's just some surface point where reactions happen. It could actually be a physical gate, or it could just be a receptor where some binding should occur that has some radius associated with it. So those are some of the problems I'm motivated by. And here's a kind of outline of my talk. I'm going to just remind you of some basic things about mathematical theory of diffusion, and I'm going to highlight specifically the role of geometry and dimension. And I'll talk about things like the recurrence of the random walk, which is important in these problems. The second part of my talk is about understanding how the organization, whether the receptors are clustered or not, whether they're uniformly covered on the surface of the cell or the nucleus, how do we understand that? Look at some homogenization limits. So how do we understand transport through membranes that have many, many, many small pores that occupy a small fraction, just like in the nuclear case? How does clustering help when you have a non-uniform collection of receptors on your surface? How does that help you form a directional signal? That's probably what's going on in mating. So once they have an initial signal, they can strengthen it by promoting receptor growth in a certain area that's already preferred. And the last part, I'll talk about nuclear dynamics and scaling. And this is kind of more recent work. So here's our friend, the heat kernel. So the standard heat equation, and here we have it in 2D, and then the answer, the heat kernel in n dimensions. So this is all kind of course, we know this, and if that was the only story, it'd be very simple. But once we put in geometries, things become a lot more complicated. And I'll talk about that in a second. I often think about the problem that a cell has is this particle here being bombarded by all these collisions all the time. And the task a cell has usually is, how do you get a concentration from collisions? So there's things bombarding you all the time. And what a cell needs to be able to do very well is get a concentration and a gradient. And that's something that they're very, very good at. It's something that humans would be really, really bad at. Suppose you were suppose you were in a room and you were blindfolded and there was a bunch of people in there and you had to diffuse around and you'd have random collisions with people. That would be quite difficult for a person to sort of estimate the density of the room just with that. And then suppose someone left the room, someone else left the room and the density went down a little bit. Could you detect that the density went down? And moreover, could you tell which door they left from? That's something that a human would find very, very difficult. What cells are doing this all the time, and they're doing it with sort of exquisite precision. So here's the sort of geometric situation that comes in. Of course, we could be in multiple dimensions, but it's applying a geometry and applying a combination of boundary conditions that bring in all the nuance. Of course, we know the standard boundary conditions that when a particle hits the surface, it could be reflected, in which case you'd have the Neumann condition. It could be absorbing, indicating that this trajectory is over, or it could be the Raw-band condition, which is a mix of the two. And that would be a semi-permeable membrane. And one interpretation, of course, is that, and it's not quite correct, but the interpretation is that if a particle meets the surface, then there's a finite probability it's absorbed and or not. Of course, this kappa parameter, in reality, if a particle hits a boundary, then it's going to interact with it infinite many times. So this kind of binomial sort of thinking is not right, but just heuristically, you can think about this. And then the kappa parameter kind of has a permeability sort of feel about it. And if kappa is very small, then of course you're mostly reflecting. And if kappa is very large, it's mostly absorbing. So let's remind ourselves about some of the challenges or some of the nuance of these diffusion problems with respect to dimension by studying these three problems. So in each case, we'll drop a particle at a distance r. And in the 1D case, we want to know, does it reach the origin? And if so, how long? In the 2D case, we want to know, does it reach a ball of radius A? And if so, how long? And the same thing, our disk of radius A. And in 3D, does it reach a sphere of radius A? And if so, how long do we expect it to take? So these are all standard kind of PDE problems. I'm teaching applied PDEs this semester, and I derive these solutions with my students. So I'll just write down the answer, of course, blah, blah, blah. But there's some things I want to just bring your attention to. So this is the solution here. And of course, the probability of capture is 1. The particle will always reach the origin. It might take a long time though. The decay rate in the tail is a -3/2. So distributions always have this curiosity that they have a very long tail. And in fact, they're simple moments like the expectation are not even finite. You can imagine trying to get the mean first passage time from this distribution. You would multiply by t, then integrate, and what you would see is that it doesn't converge. So these distributions are always difficult to work with in the sense that you can expect extremely long events. The capture time can be very, very long, even though it's always guaranteed. And as you move up in dimension, basically Brownian motion is a less effective mechanism for exploring space in higher dimension, because as the volume you have to explore increases as the dimensionality grows, you're still limited by your mean squared displacement as a function of the square root of time. So diffusion is just a weaker method of exploration in higher dimensions. And the critical dimension is 2. So we look at the same problem here in dimension 2. The analysis is a little bit trickier. Here's the solution. It's a little bit gnarlier, but it's the same. It's kind of similar in the sense that the probability of capture is still 1. You're still guaranteed to enter that disk of radius A. Now, here's the problem that all calculus students hate, of course. 2 over t log t squared. So this converges, but it's right on that boundary. So it's that 1 over, does 1 over n converge? No. 1 over n log n. So it's that kind of right on the border of convergence, this distribution. And then when we go to 3D and ask the same question, then of course, then you're no longer guaranteed to hit the target. So if you start at R and ask when do I enter this ball of radius A, you're no longer guaranteed anymore. And actually that should be A over R, not R over A. The probability decays monotonically here. And yeah, that should be A over R. And what this is basically saying is that if you're 100 radius away, your chance of hitting it is 1%. Now, if you do hit it, then you still have this long tail. So you still have the long tail phenomena, and that particles that do reach there will take a very long time to get there. So that is the recurrence of the random walk. And an important thing is that even when you do get there, your actual capture time can be very long. So let's look at this scenario now. What about in this more biological scenario where you're not just hitting a uniform target? It could be a cell with receptors on it. Now, if you think geometry is important and that the cells are obviously some manifold, some membrane, then you can account for that. You can also look at the reduced problem where they're just receptors on a plane and ask when, you can ask about the arrival times there. So what I want to tell you about is how myself and others have sought to find the statistics of these events. So actually, okay, these slides are in the wrong order. Okay, so let's look first at just the probability of capture. So I'm going to tell you about this Berg-Purcell result, which was a famous result actually back from 1977, a very highly cited paper. So let's just start with a cell here, and then there's the receptors on here, and ask, well, what's the probability the particles captured? You can also think about U as being a chemical concentration here. And from the divergence theorem, you get this formula that the flux, that's the flux into those receptors, well, it's the diffusivity multiplied by, and you just sum up the gradient over each of the pore surfaces. So this omega A here is the combined surface receptors. And that's 4 pi times the diffusivity times this number called the capacitance, which is kind of, capacitance comes from an electrostatics kind of nomenclature. So this is also a really important problem in electrostatics. You look at these, this is like an insulating sphere with charged electrodes here, and you look at the electric field outside and C is the capacitance. So it's basically the decay rate at infinity. So if you look naively at this, J is, okay, multiplied by the diffusivity times this integral. Naively, you might think this integral should scale like the area, because you're integrating over the area of the pores, so or the receptors, so it might scale like the area, but in fact, it scales like the perimeter. So this was a really big result that Berg and Purcell showed, and they kind of did this informally, that if there's n small non-overlapping receptors or pores, and they all have a common radius sigma, then they kind of derived this formula, and they did it by interpolating between two limits that they thought that they knew were important. Well, if it's all covered, it should be this. And if there's just a few, it should be this. And they got the leading order formula here. So it's 4 times D times N times sigma. And why is this a really important result? Well, it shows that the flux is proportional to the perimeter of the pores, not their area. That's very important, because if you're able to devote a small fraction of your surface area to receptors to a certain antigen, then if you subdivide those, your area stays the same. So in this configuration here and this configuration here, the area is equal, but the one on the right has a much larger perimeter. So if you subdivide a circle in two, its perimeter increases. So that's kind of one of the main reasons why cells can be very efficient at detecting their environment and be specialized to all sorts of different molecules with only a small surface area fraction devoted to that. However, there's no information here about the spatial arrangement of receptors. This can't tell you anything about clustering and why clustering might be important. But let me give you an explanation for this important result. Now, if we zoom in on a single pore and we ignore geometry, you can imagine this sort of caricature problem. Just imagine a single pore, single receptor in an infinite plane that's reflecting. So you have to solve the Laplace equation with 1 on this little disk and then no flux everywhere else. You can solve this. It's kind of a classic problem in integral equations. And what you find is the flux is singular. So if you look at the surface flux, of course, it's 0 exterior to the cell, but interior, it's got a vertical asymptote as r goes to 1. And it's an integral singularity. And you can see the type of singularity here. It's a 1 over the square root of 1 minus r squared. So there's an integral singularity at the meeting of Dirichlet and the Neumann part. So where these boundary conditions meet up, there's a singularity. That's another thing that's challenging about these mixed boundary value problems, is that if you have some sort of surface and you want to apply boundary conditions to it, if you mix and match the type, if you start picking, hey, I want some Neumann here, I want some Dirichlet here, I want the Robin condition here. When you do that, you get a singularity at the join points. And that's the reason for this fundamental result. And it also is it's a note of caution for when you're doing numerics, because your solution is singular and you have to be very careful about evaluating the solution under those circumstances. But here you can see that kind of the most likely place to be absorbed. Suppose there's a particle out in infinity and it was going to be absorbed in here. Where is the most likely place for it to be absorbed? Or sorry, the least likely place for it to be absorbed is right at the center. And there's a sort of analogous physical process. Like actually, the physics of this are completely different. But you imagine rain going down this drain, and yes, some raindrops are will be falling directly into the drain, but most of the flux through this drain is coming through the surface. And that's kind of why, that's what's going on here. And that's why the flux into some set of receptors depends on the perimeter, not the area. Of course, the physics in this situation is quite different. So going forward, how do we understand clustering? Well, it's quite tricky actually, because a lot of the methods we know for solving PDEs are actually devoted to either just got Neumann, just got Dirichlet. There's actually some simple problems become extraordinarily complicated once you start mixing matching the boundary conditions. And what a lot of people have done since then is gone to this boundary homogenization strategy where let's try and ignore those surface details and just replace this mixture of Neumann and Dirichlet with a uniform Robin condition, if you can choose the kappa appropriately. So how should this kappa be chosen? Well, obviously it depends on the receptor configuration and how absorbing the surface is, how the absorbing fraction. And what's what's kind of the state of the art a little bit is kind of fitting to some functions that mimic the properties that we know. So here F is the absorbing fraction. It's the fraction of the surface that is absorbing. Sigma is the individual receptor radius. And what you can do and what people have done is fit this to certain functional forms that they think fit all the needs. For example, it should diverge, and we think that, and I've got some reasoning why it should diverge like 1 over 1 minus f squared as f goes to 1. So this is kind of the state of the art up until a few years ago. So what we were able to do is actually go back to the original Berg and Purcell problem, and this is, I guess, back 4 years ago now, and go back and from first principles solve that problem again. And we were able to get the Berg-Purcell result with the additional terms that tell you how the configuration of the surface receptors influences the flux into the cell, and therefore some proxy of its sensing ability. And what you see is the following. At the beginning, you have the classic Berg-Purcell result. Then you have the effects of spherical geometry. So the fact that you're on a sphere rather than something flat. And then after that, you have the competition effect between the pores and the receptor or the receptors. What is the competition effect? Well, if pores are competing for flux, if a particle is coming into the cell, it's going to go to one of the receptors, and if the receptors are close together, then they're kind of competing for flux in a sense. And this term here accounts for that. It looks similar to a kind of Thompson problem. So the Thompson problem is a very classical problem in classical physics of how do you find the minimum energy configuration of n charges on a sphere. So if you take n electrons, of course, they want to repel one another if they're very close, but if you constrain them to sit on a sphere, they want to be mutually far apart from one another. The Thompson potential is just 1 over d. But it's a problem that's well known to have lots of minimizing configurations and very, very difficult to find the global minimizing configuration. Now, the interaction kernel itself, if you plot it, it's decreasing, it's concave up. And that kind of confirms this intuition that, look, they want to be as far away from each other as they can. They should be equidistributed. So there's not some preferred distance. What this means is that if you want to maximize flux, you want to have a homogeneous receptor covering. So that's what this is telling us. To maximize the flux and to minimize the competition for flux, try and make them all as far away from each other as you can. Now, how do you get a covering of this cell that's uniform? Well, If you just choose points randomly on a sphere, you actually do a really bad job of that. So here on the left shows points randomly on the sphere. And what happens is you see them clustering. So there's a little cluster here, there's kind of clustering here. There's open spaces here, which is not good. So this is actually a very poor homogeneous covering. That's just uniform random. So it really does not do a good job. Now in spherical coordinates, you can just take the spherical coordinate system and look at equal space there, and that does a better job. But a really nice point is the Fibonacci spirals, Fibonacci spiral points, which is a grid given by the following algorithm here. That if you look at it from the North Pole down, it just does a big spiral. It's a very nice, simple covering of the sphere, which is very nicely distributed. There's actually been quite a lot of effort gone into developing these coverings because they're very important in meteorology. And the reason is that meteorologists would like to do very fast integrals over a sphere like the Earth. And that can be done very quickly just by a naive sum if you can make each little box have equal area. So by having a very nice, easy grid that's equidistributed, integration becomes trivial. You just have to sum everything up. It's also very important in satellite when they're mapping out the sky for like CMB or something like that. They want to have a nice grid in the sky to point the satellite at. So there's a lot of work on optimal coverings of spheres and other manifolds. What about homogenization? Suppose there's many particles. So we started with n. And the point here is to try and take this sum and replace it by an integral. That's how you do homogenize. That's one way to do homogenization. Just saying that, well, as n goes to infinity, as the number of particles here goes to infinity, this should really be replaced by an integral. So what you do here is you take the number of particles going to infinity and the individual receptor radius going to 0, and you keep the surface area fraction fixed. And if you do that, you can derive the following kappa here. So this kappa is nice because it's coming from first principles. It's not like a theoretical result, but it is coming from first principles, and it lets us know what all the terms in here do. The first term is Berg-Purcell, that classical result. The next term, the square root, of course, it's got the negative sign on it, indicating that it's decreasing the flux. Actually, it's all in the reciprocal, so I've got to be careful there. But this term is the receptor competition. We have the logarithmic term, which describes spherical geometry. Now, if you're wondering where that, maybe I didn't explain this actually, I can give you a little bit of intuition. Why is there a log term associated with spherical geometry? The reason is that if you recall, the Green's function in 3D is a 1 over r, and in 2D, it's a log r. The log term comes in because there's an element of 2D diffusion. Once the particle hits the surface and then diffuses on the surface, it's kind of combining a 3D diffusion with a 2D diffusion. And that's why the logarithmic term is there, because essentially this result combines the 3D search to get this onto the sphere, and then when you're on the sphere, you do a planar search to find the receptor. So it's a combination of those two. That's what that term's all about. Let me actually go to this. So let me just say one slide about the numerics. I'm not sure if people are interested in numerics here, but again, one of the challenges is that this solution is singular. You want to calculate this solution, but it's singular. So you have to be extremely careful about how you go about this. And the way we decided to do this was with integral equation method, specifically the Neumann to Dirichlet map. So if you know the surface flux and the Green's function, you can map it to the surface potential. So what we did is we took this integral equation and then we used a spectral expansion in terms of a basis function that we knew mimicked the known singularity. And that was allowed us to solve this with spectral accuracy. And what we were able to do is verify this formula to show that this formula does quite a good job representing the flux. And for 1,000 particles, we were able to show that it's within about 0.3% of the relative error was about 0.3%. So we have some numerical validation methods for showing that this could work. Recently, Leslie Greengard and his student Jason Kay, they extended the method using fast multipole expansions. You can imagine, I can tell you a little bit. I mean, you basically have a little local basis near each pore, and then you have to do this integration. So what is the effect of basis I on basis J associated with some other pore? You have to do all these integrals. The fast multipole method is a way to accelerate that process. And they were able to take that and then go all the way up to 100,000 pores or receptors, which is really, that's where you want to be in terms of some biological limits. So, so this can be extended to extremely large number of receptors. Here's a neat thing as well. So, of course, we just looked at the capture time, the capture likelihood, the possibility of capture. Now, what about the arrival time? So if you take the kappa we derived from the steady state analysis, substitute it into the heat equation, this is an exactly solvable problem. And then you compare with the actual arrival times. Again, I want to draw your attention to the extremely long arrival times here. This is time on the horizontal, 10 to the 2. So you really, really have a very fat tail in the distribution here, just plotted on a logarithmic scale. What you see, surprisingly, is that the homogenization of the steady state does a very good job of predicting the flux in the fully time-dependent problem. And perhaps that's not too surprising, and that's basically the saying that, look, the explanation for this is that if you're interested in the full distribution, which means large times, then there's only so long that the initial condition has a very large importance. So very quickly you lose initial information, and then afterwards you're just interested in more of an equilibrated problem. And it's not so surprising that the homogenized density does a good job. So here's a representation of the sphere here that we use in our computational simulations. And in this case, what you're seeing here is 51 receptors, and they have a 10% coverage fraction. Let me just show you a little bit of a simpler scenario. Things are a little bit simpler on the plane. So I remember we talked about how, well, do you think the geometry of the cell is important? All right, well, you can put it on a surface. If you don't, just put them on a plane. And then you have fewer problems because you can define a periodic structure and just repeat that. So here, for example, if we define two linearly independent vectors, then we get this what's called the Brevis Lattice. And it's basically a tiling pattern for the plane. So here, for example, I'm showing P equals 1/3 1, and then Q is 1 0. And this is the plane you get. This is the tiling pattern you get. And you can do other things, like you can look at perturbations like this, like almost periodic Brevis lattices, and we do have some results on heterogeneous lattices, so that inside each lattice it's not all the same. But this is the basic framework, and what we're able to do is go through a similar process and get the homogenization parameter. And here it is, kappa. Now, like I said before, this result is specialized when all the pores are the same, but you can go back and do it for if the pores are different. And in fact, you get something like C bar is the pore average, but it's the capacitance. It's just a number associated with the shape and the geometry of each pore or receptor. But nonetheless, here's what you get: a much more compact formula, because you don't have that logarithmic term, which is arising from the spherical geometry. Here you get this term here. Now this function R, it's known as a regular part of the Green's function. So remember, the Green's function has a singular component and it has a regular component. The R comes from the regular part of that. It's something we can write down a separable solution for. The separable solution does not have the greatest convergence properties, so you have to do some manipulations to it to make it converge quicker, and you can do that. Essentially, you can write down an infinite sum for this. And since a Brevis lattice is defined by two independent vectors, that's ostensibly four free parameters. You can do some tricks though. Why don't we make the repeating cell unit area, and without loss of generality, we can put one of the independent vectors. Just let that be on the x-axis. So we can get it down to 2 parameters. In fact, it's actually, many people have said this, why don't you just use polar coordinates? And polar coordinates is much more convenient. And I think we kept with a Cartesian formulation because we like the picture this produced. So with those 2 parameter situation, it's quite easy to scan over the whole family of Brevis lattices. You can just scan over every single potential lattice and therefore get a picture of all the potential configurations that are maxima or that are critical points. And in fact, you find there's 2 critical points only. So this landscape has 2 critical points. One of them is the hexagonal lattice, and that's the global maximum. That's the one that maximizes flux. Now, this is very, very common in a whole bunch of pattern forming systems. So this is not too surprising. There's many, many pattern forming systems that where the hexagonal lattice is some kind of minimizer or critical point or maximizer. The square lattice is also in there, and it's a saddle point. Now, in this picture, you see all kind of the opaque region here is just repeating of this area. This is a fundamental cell here, and everything else is just like a reflection of it or mapping in the hyperbolic plane. So it all just repeats down here, but more intricately. And this is really what you're seeing here is it's a manifestation of the parameterization that we took. I think we just did this naively in a Cartesian form and we thought the picture was cool, so we kept with it. But it actually looks a lot more sensible if you use a polar representation of the Brevis vectors and you just look at an angle and the angle and the radius. Okay, so that's what's happened with the planars. The planars are a little bit simpler in some sense because you don't have to deal with that geometry. So let me talk a little bit in part 3 about how to what we've been working on with regards to clustering and directional sensing. So here's the kind of basic framework, and it's similar, of course, to what we've been talking about, is that now it's sort of an inverse problem where now this cell has information at its receptors, it has information at its surface receptors, and it has to try and understand where the source is. This is work with Sean Lawley at Utah and Chris Miles at Cron. So it's a kind of an inverse problem. So the idea is, given the arrival information of a finite number of particles at the surface receptors, is there a way to build a distribution or an understanding of where the location was? This is also kind of similar to gradient sensing as well, where the cell has basically got to establish a gradient from the distribution of arrivals on its surface. So we took a sort of information theory framework to tackle this. This is a problem that's been tackled by many, many folk. And one of the things that's been of interest to many researchers is the amplification factor. So what cells, as I alluded to earlier, are incredibly sensitive, and that basically what they can do is, if there's thousands and thousands of receptor signaling molecules instant on the receptors, they can essentially tell from a difference of single digits between the front and the back, they can get a gradient from that. So there could be like 4 more arrive on the back than arrive at the front. And that's enough for them to get a very strong signal with all the noisy circumstances. So there's a lot of research. It's like, how does that tiny signal get amplified so strongly? Whereas this portion of our, I'm talking about the very initial part. And of course, the understanding is that some downstream mechanism, some processing of the signal, once it's actually reached the receptor, something downstream is contributing to that. What I'm going to talk about here is purely the receptor organization. So, and of course, in the case of yeast, in some cells, they're uniform. In other cells, they will actually dynamically reconfigure the receptors once they've received a transient signal. So as I was mentioning before, we took a sort of information theory sort of perspective on this. And one of the things we introduced is, one of the things we have here is the Kullback-Leibler divergence. And what this is really a metric that allows you to look at the difference between two distributions. So it's the information content gain from revising a distribution from Q to P. Now, let's look at this situation here. Let's look at this situation. On the left, what we've got is a scenario where here's the cell, and particles have diffused and they've arrived on the cell surface. And these arrival sites have been drawn from a uniform distribution. So they're uniformly spread all over the cell. In the case on the right, the locations on the cell surface where the particles have been incident are drawn from a distribution where it's above the North Pole at a distance r. And you can see just in the eyeball norm here that, of course, there's way more particles that have arrived on the North Pole than have arrived on the South Pole. Hopefully that's clear there. That, I mean, I think there's like maybe 3 particles in the South Pole where the majority of them are on the North Pole. So therefore, there's definitely a signal there. Hey, if I'm going to guess, is the source above me or below me? You're definitely going to say it's above. Okay, so this is an exact distribution. So if there's a source at a distance r above the North Pole, then this is the probability distribution that will be incident on the sphere. So one of the things we can do is calculate a kind of theoretical bound on what's the most deviation from uniform that you can get. So this is perfect in the sense that there's infinite number of receptors, so that wherever the particle lands on the cell, it acknowledges that location. And there's an infinite number of incident particles as well, so that you have continuous distribution. And then you compare, like, okay, how much information content do you get compared to uniform? So we evaluate this KL divergence, get this, and you can get this exact formula, which is just a function of r, the distance. And you get something that's kind of intuitively makes sense. As r goes to 1, this diverges, so that, look, if the target is really close, you get a lot of information. If the target is really far away, you get less, and that decay rate looks like r to the negative 2. So there's something interesting in here. So, and one of the things that people have noticed with these sort of problems, that cells are able to get a gradient, not just if they have a very small gradient, but when the source is many hundreds, even thousands of radii away. So the decay rate r to the negative 2, okay, it gets small, but it's not getting crazy small. It's not getting exponentially small or something like that. So it gives you a sort of, what is the best you can do? So it's an upper bound on what's possible. And now we want to see how a cell can approach that upper bound in a finite receptor scenario and in a finite data scenario. So Y here is just meant to reflect a configuration of receptors. So receptors have locations and they have radii. What is PK? Is the splitting probability. It's the probability of absorption at receptor K. I've got a question here.
Yeah, we have a question from Zhang, so let me just unmute her.
Oh yeah, no, I see it. Yes. Yep. Yeah, no. The question is that the KL is not symmetric. So how do you decide which is Q and which is P? Yep. We used Q is in the uninformed or the uniform direction, because that's what you start uninformed. So your idea is that you start uninformed. As you receive signal, then over the receptors, then you update. So your previous is always uniform. You're starting out from uniform, which is being initially uninformed. Does that clarify? Okay. Okay.
Yeah, I think she.
Is that okay?
I think she's fine with it. Okay. Yeah. Thank you.
So the splitting. Okay. So the splitting probabilities are, okay, what's the chance you landed on the kth receptor? Those are called the splitting probabilities, and we can work them out. We can do it analytically, or sorry, asymptotic limit, we can do that, or we have a full numerical solution that allows us to do that. So we've got both capabilities. This asymptotic solution is valid when the receptors are well spaced and such. So highly clustered, this is doing less well, but we have a full numerical solution that can do it as well. And we've tested with both. Obviously, this is much, much faster. The asymptotics is very, very much faster than the full numerical solution. Okay, so we took that theoretical max, and then now we're going to look at finite receptors. Previously, there was infinite receptors, so that wherever the particle landed on the surface of the cell, it was instantly recognized. Now it has to land on a specific location. And we did two experiments on this. First, we fixed the radius and then varied the surface coverage fraction. We also fixed the surface coverage fraction and then varied the radius. And in both cases, we do see that you can approach this theoretical maximum limit. So we looked at the ratio of what's the KL for this finite receptor case versus the ultimate case. And you do approach it. You do need quite a lot of receptors to get really, really close, but you can get there. And again, of course, you have this r to the -1/2 decay. So the point here is that with a finite number of receptors, you can get close to theoretical maximum performance. Now, what about specifically with clustering? Okay, so this, I will say, is I forgot to mention, is this is based on a uniform or a homogeneous covering of receptors. And well, what about clustering? So here's two scenarios. One where you have uniform clustering, and the configuration on the right, configuration on the left is uniform receptors. Configuration on the right is clustered receptors. And then we look at the information content. And remember, this is the directional information in the sense that it's what gives you the largest deviation from uniform, what gives you the strongest signal. And what you see is that the uniform gives you a smaller deviation. It's just smaller peaks, smaller troughs. The clustered, maybe not surprisingly, has higher peaks, higher troughs. And if you look at the difference between the two, what you see is that you've generated some potential where there's regions that if the cell is right in front of a cluster, then you can do better. And if it's not, you might do worse. So this suggests that, look, if you can align a cluster towards a target or a source of interest, then this is a mechanism that can allow you to increase the information content over the receptors. This effect is less important at larger R. So smaller R, you see an effect that's quite pronounced. As the source distance increases, things become flatter and flatter, and as the configuration of receptors is less important. So this suggests a possible mechanism at close range where alignment can improve your information content. What about practical recovery? We used a MLE method to do this. So what about practical recovery of a source? Well, if you have the count data so the CK here is the count at the kth receptor. There's n receptors, then you have m total particles. So you have m particles incident on your cell, and they're distributed amongst the receptors, of course. This is a multinomial distribution. And if we look at the log likelihood of this multinomial, it's the following form here, where it's the sum over the counts divided by m. That's the fraction that went into each receptor. CK over m is the fraction. It's like the splitting probability multiplied by the log of the splitting probability. And this is actually, there's a subtle point here. This is the conditioned splitting probability. Because remember, in 3D, you don't have recurrence, so some particles will never even reach there. So it's a condition on the fact that the particles do arrive. And this is something that you can look for its critical points. Okay, so we can take a rough estimate by looking at the argmax of this. And we see something that you might expect is that if you get more data, and data is going in the horizontal here, then you can get a better estimate of where the source is. And as the source distance increases, this becomes tougher, and then your estimate has more error in it. Now, I have to really emphasize something, that we're not suggesting that the cell does MLE, although there is, some people do argue that. It's just a mechanism that we're using. Given the count data, it's just a mechanism we're using to go directly to some inference for the source location. So what's the point here? What's the sort of finishing slide here? It's that, look, if you have a homogeneous covering, you're maximizing flux. However, if you go to a clustered configuration, you take a flux penalty, roughly 5% here. The flux is roughly 5%, but there's a potential that you can become more sensitive to the source of the particle, to the source. Yes, that's the sort of trade-off you pay. Homogeneous coverings maximize flux, but you can get an advantage in directional sensing if you have a clustered configuration of receptors. So that's part 3. Let me talk quickly about part 4, which is some of my newer research. So let me actually show a video first, maybe if I'll do this. And this is data from experimental collaborators, Jay Gatlin at University of Wyoming. And what I want to show you here is this video. Can everyone see the video okay? Can you see that okay? Can you see this PowerPoint?
Yeah.
So what you're seeing here is a frog embryo. And what the Gatlin lab is able to do is tags GFP NLS, which is associated with import of the nuclear membrane. So when I play this video, what you're seeing here is over the course of maybe an hour, the growth of the nucleus in these cells. So these are embryos of frogs, and this is the nucleus. It starts small and it grows large. This is an interesting cell here in that it has 2 nuclei in it. And you can see there's some interesting dynamics where they're processing here. Often they seem to start on the boundary and then move to the center. So this is a bit of an anomaly here. So here's the sort of picture of what's going on, what these guys are able to do. Here's those snapshots here. Well, there's this, they can tag this and image this GFP NLS, green fluorescent protein, nuclear localization signal. And what this allows Jay to do is figure out the import rates by looking at the illumination. And of course, you can just measure from the photos the cross-sectional area, but you can also measure from the light intensity, you can measure the import rates of material over the nuclear membrane during growth. So what we'd like to be able to do is answer something about the scaling here. What mechanisms cause the nucleus to have the volume that it does and the position it does inside the embryo? So what you see on the right here, so the red is if you're looking at a cell with no embryo, then GFP NLS intensity is just constant. There's no nucleus, it's just fixed, it's just constant. And then when there is a nucleus, then the GFP NLS decreases, cytoplasmic GFP decreases, and the surface area of the cell increases and equilibrates. So what we have here is very good time course data on the dynamics of the import rate and the extent of the nucleus in this model system. Actually, I'm not in the event here. And what we're working on right now is building a mathematical model to explain this. So let me tell you the sort of 3 key components very quickly. I know I'm getting close in time here. So what's the general situation? Well, assembled materials have to be transported to the nucleus. That's by unbiased diffusion and advection along microtubules. And here you can see those visualized here, the microtubule network for a single nucleus and 2 nuclei. Then once they reach the nucleus, they've got to be imported over the nuclear pore complexes. And we know the size of those is roughly 40 nanometers, and we can count them up and such. And then somehow that imported material is converted to growth when the nucleus grows, and we have very good time course data on that growth. So we want to build a model that tries to reflect each of these parts, and actually the most complicated part, part C. So let me just tell you a little bit about the models we're building. And okay, we're going to ignore advection right now. Of course, that's very important. But for now, just look at diffusion in an annulus or a concentric spheres. So as you saw in those pictures, the nucleus tends to start on the boundary and then drifts towards the center. But we're just starting at the center. Of course, there's no flux on the boundary. And on the nucleus, you have some import here. And here the kappa is the permeability parameter that we derived earlier in the first part of the talk. It tells us, like, if you know the MPC radius and you know the fraction of MPCs on the surface, then this is your permeability. And what we can do is derive, just from some basic integration here, the total signal that's input over the nucleus. Just the flux in here, of course, it's the surface area times the permeability times the local signal. And what we're working on right now is trying to understand how imported signal is converted into radial extent. So for example, is imported signal converted to radius, area, or volume? So if you take in some building blocks, does that allow you, does the radius scale, the area scale with that imported signal, or the volume scale? So we have three hypotheses here on what might happen. Are NPCs created during growth? Well, if there's constant, if they are, then the permeability might be fixed as it grows, indicating that more are being added to the surface. If it's not, if there's a fixed NPC number, and then as the radius grows, the permeability is decreasing because there's only so many gates on the surface, then that would be hypothesis 8.2. And what we're doing is using this modeling framework and some statistical methods to sort of either give evidence for, using the data to give evidence for, or evidence against each of these hypotheses. So for example, if you compare these hypotheses all together, the data we have so far, which is very nicely calibrated to our modeling, suggests that volume scaling is the most accurate, in the sense that any imported material gets converted to volumetric extent. And the H2 in that we have, did I get this wrong? Sorry, I mixed this up here, that we have constant NPC density. So that suggests that new NPCs are being constructed as the nucleus expands. So this is some preliminary results we're working on and hoping to get this wrapped up soon. So let me just end there and just give you some concluding thoughts. And basically what we've been working on the last few years is a whole bunch of techniques to try and understand diffusion through complex geometries with multiple sites and heterogeneous membranes. I didn't talk so much about the numerics, but I've spent quite a bit of time on the numerical methods for doing this as well. And some of the important things is like that we've seen is that, look, if you want to maximize the flux, then the receptors should be as far away as possible. That doesn't happen randomly. There must be a mechanism to do that, by the way. So if you think that if you just throw a bunch of darts on the wall, they will cluster. So if you see some sort of structure where there's a homogeneous covering so that they're equally spaced, then there has to be a mechanism responsible for that was worthwhile to establish so that you would increase your flux. We see that in muscle cells inside with multinucleated cells that have multiple nuclei, that they're spread out in order to sort of equidistribute their area. And there's a mechanism that performs that because it's important to make sure that the fluxes are well spread out. Homogenization, we can replace all the complex surface data with macro laws, and we can do it in a systematic way that's verifiable. So you don't have to have a crazy mesh to resolve all 100,000 pores or something like that. You don't have to do that. You can just use these homogenized formulas and they work. With regards to directional sensing, you can take a flux penalty and you can make yourself more able, the cell can be more able to get more information from directional sources. Okay, so let me finish there and just highlight some papers that this work is drawn from and acknowledge support from NSF. And thank you all for your attention. Thank you.
Thank you very much for such an interesting talk. So please feel free to ask Alan questions if you have any.
Thanks, Alan. That was really a very interesting talk. One thing that I wanted to ask, so when you showed that image of the nuclear pore complexes kind of spread out over the nucleus, do you see this sort of uniform distribution? Does it appear that indeed they're trying to, there's some pressure to maximize the flux?
You know, this is really interesting, and I think that's actually quite difficult as well. It's okay, when I look at it in my eyeball norm, I think yes. But I've been to some talks where they're like, look, this is really difficult because these images are of a three-dimensional surface that's being squished under a microscope. And actually the basic problem of can you even get the XYZ center data is not easy. So it's actually super difficult to really statistically show that there is that it's not uniform.
I see.
That it's not like just random, that it actually is equidistributed. So I don't think it's so simple. I don't think it's so simple.
I guess that could give some indication of whether this.
What's actually really difficult is to actually come up with a synthetic. I think would be really interesting is and challenging is to be able to come up with synthetic receptor configurations that actually mimic what's seen in, because some of the things I've done here are, maybe you can see how I've cooked up this little, I took a little pattern and I repeated it over the sphere, but in actual fact, there's way more structure in the real receptors, and usually it appears that certain types are put together in little microclusters, so that repeat engagements become easier, and with different types of ligands as well. So there's a whole bunch of stuff that's going to get swept under the rug here. But first of all, looking at a microscope image, and without knowing the surface, like you don't know what the surface is, you just have a 2D planar image. Reconstructing the XYZ coordinates of the receptor centers is not easy.
And I think, Janne, if you want to go ahead and unmute yourself, you can also ask any questions.
Yeah, thanks. Yeah, hey, that was a really nice talk. Thanks, very fun. Yeah, no, I was just curious about the stuff you were showing at the end on the growth of the nucleus. So, do you have terms, or have you thought about sort of terms that might limit the growth, so so that you kind of reach a stable nuclear size through that kinds of dynamics that you're describing?
Yeah. Yes, our model. Okay, so our model as it is right now, and also this is preliminary, it can reach equilibrium, but not until all the material in the cytoplasm is converted into nuclear envelope. Right.
So your idea is that it's a finite pool of whatever the limiting material is that's controlling the size of the nucleus.
Yeah, and that's where we're operating right now, but it's not.
And presumably you don't know which particular molecular species this would be that's limiting the growth, right?
Exactly. So if you look at this data set here, you can see that in these characteristics, they have almost depleted. Well, actually, sorry, they've gone down to about 10%. So it's gone down from 10% of start. This is kind of, this curve here is the material left in the cytoplasm. It's not zero, actually. It's, which may indicate that there is another mechanism that's causing equilibrium. This is really
and sorry to interrupt. What's labeled here? What's GFP on?
GFP NLS is a green fluorescent protein. NLS is nuclear localization signal. It's
Yeah, but that can go on any protein, so I'm kind of confused a little bit. Right, nuclear localization signals are, as far as I know, are small peptides that are attached to proteins that are important to the nucleus. So that's
Right.
not specific to any molecular species. That's just labeling everything that's targeted for the nucleus, right?
I think that's what they're doing here. I think they're just labeling everything. That's what they're
Wow.
that's what they're.
That's
I mean,
that's
this
an
is kind of a very
interesting.
controlled environment. This is so they can control exactly what goes into these droplets.
So these are droplets filled with extract.
Exactly. Yeah.
Yeah. So have you looked at sort of older data from Rebecca Heald's group that also did these? I'm just curious whether the sort of growth that you see here is the same that they were seeing in Xenopus extract that was like maybe 10 years ago.
Who did you say there? What was the name?
So it's Rebecca Heald. She's at Berkeley. She's the one that started this whole business about 10, 15
Okay,
years
I'm
ago.
gonna have a look at that. Okay, I haven't.
Yeah, I'm sure your collaborator would know about the work. In fact,
sure.
I suspect that he or she is out of that lab because this is very classic sort of.
Yes.
Okay.
So, Alan, several of us here at Brandeis are really interested in these questions of size control, and really, Yane Kamdev is the guy who's
right.
the one to talk to. So yeah, I think especially with these limiting pool models.
Yeah, you're right. We have we're working on some more sophisticated models right now, but our model right now is a limiting pool model. But what effects are we looking at? We're looking at mechanical effects on the membrane, so bending forces, oncotic pressure. When we're looking later on at microtubule effects as well. So that's what we're kind of, those are the things we're.
And do you have a sense that the advective terms from the microtubules will be kind of competitive to the diffusional terms that you've included in this first attempt? I mean, like, will it be a significant contribution? Do you have a sense of the size of that?
I mean, yes, I would say yes.
You think so?
Yeah.
Great.
No,
Thanks a lot. Yeah.
Yeah.
Super fun. I don't want to monopolize your time, but really cool stuff. Thanks.
Oh, thanks. And I'm sorry I mispronounced your name. I missed.
No. I'm Jane. Everyone calls me Jane. It's all good.
Okay.
Yeah, my cousins always make fun of me. Don't worry about it. Good. All good. All good.
All right. Do we have any other questions for Alan? All right, I guess we'll just wrap up here for today. I'd like to thank our speaker, Alan, again, and everyone for participating. I will hope to see everyone again next week.
Thanks, Alan.
Thanks very much. Thank you, everyone. Have a good one. Bye.